Good morning, all. Today, I have come up with a new topic called what do we say, Pharmacophore modeling using web servers. So, let us start with the existing Pharmacophore features from in my last video. I have shown you how to perform Pharmacophore using Zinc Pharma. So, now let us go with search Zinc. So, here I'm just loading the receptor. So my receptor, I'm selecting 4KTF and I'm just clicking open. So now my receptor will be present in my, what do you say, this one, what do you say, key on the workbench or then on the, what do you say, web server. Now this is my receptor, which is having an inbuilt ligand. Now you need to specify the pharmacophore features which you want to search the library. Just start to add features. So I'm keep on adding 3 features. At least 3 features are necessary for run a pharmacophore search. So now I'll just go for the 4 features. Now I don't know exactly what exactly is my pharmacophore, the where the ligand is binding, I do not know. So this feature is essential in performing any pharmacophore search. So here I'm just clicking on, without having any knowledge, I'm just clicking the basic features and I'm just submitting the query. Once I submit query, you can see it directly says that there is no hit or found. So now let us play with another type of search where I am minimize the pharmacophore by just selecting the coordinates of my binding pocket that is very to the shorter length. So now as the auto pharmacophore feature is not available in this server, so you are facing some issues in identifying your what do you say, the pharmacophoric hits. So you should also remember one thing: you should not be greedy enough and increase as much as features in your data. So as you increase the number of pharmacophoric features, so the chances of rejection of your pharmacophore search happens. Now see, earlier I have given coordinates to the 10. Now I have reduced the coordinates to the 5. Now I am submitting my query. Still I have not got anything. So let us remove one of the features, and this feature I will remove. So big, oh sorry, all are hydrophobic. This is what is the error which I have done. Let us change it to all the features should be aromatic, and this is one should be hydrogen donor, and one will be hydrogen acceptor. So now this should be different in each other, and now you submit your query and check how it is, how it helping. Now you see still you do not find any hits. So further you can even change it to 7, and further you can try how your pharmacophore helps in this. And again you see further whether it is helping you to identify things or not, and you just check, and again you submit the query, you check what happens. So, and there should be some logic enough in doing such type of searches. So let us fit some method. This search which I am showing you has no logic, because I am not inputting any kind of features in a perfect mode. So now I will just show you the different server which has an auto pharmacophore feature. So how it will help you to identify the pharmacophore directly. So now here I'll just click on to this I'll just go with 4KTF. So this is my ligand, and I'll just click on submit. Now once I submit, my pharmacophore will be directly present on the screen. So this is my protein, and just wait. So this is my ligand and pharmacophore. Earlier, before submitting, it is showing other 2 parts, that is heme, metal and sulfate. These are the cofactors. This should not be selected as a ligand. Once you select these as a ligand, your basic pharmacophore search is itself is wrong. So now if you input these coordinates into your Zinc database, it will perfectly provide you the results of that that search. So here, which you are failing to identify the search, here you are able to identify the search automatically. Now you can even search zinc on this. Once you click onto the search mole prot, it is searching around 64 lakhs 8,824 and 361 molecules in the database, and it is presenting these many hits. That is 17,471 hits are available on this molecule. Now let us click any one and check whether such molecule is having similar structure with our structure or not. So now this is my protein target, which is having a binding pocket. So now we are going to an conformer space. Now see, this is your structure, which is uh, submit, which is searched in your database. So now you see the structure is also almost similar to your structure which you are already presented here. So this is the beauty of this software, and you can have multiple what do you say, you can have different types of servers, what do you say, databases, and there you can identify n number of what do you say molecules in your hits. So as you increase the future, suppose let us go with increasing the futures. I am completely increasing the futures of my pharmacophore, and if I am clicking hit, so what happens as the number of futures increases and the total now if I search, I may not even get a single hit because I was more greedy to get the exact fit molecule. So now I may get 0 or I may get 1. So selection of pharmacophore futures, selection, you should have a logic. See, earlier we have 17,000 hits. We now currently we do not have any hits because we have made these futures are vast and we are more greedy in identifying the data. So this is what you should careful and logical in attempting any pharmacophore model. So now pharmacophore, there are two types of pharmacophore, that is a ligand based pharmacophore and a structure based pharmacophore. What is ligand based pharmacophore? Suppose you are targeting a protein structure which is already reported in the literature, which has certain 100 to 200 ligand molecules which are active and which are present in the database. Now you will take those 100 molecules and create an ensemble, and that is superimpose all these molecules at one place and create a pharmacophore, common pharmacophore feature based upon the ligand attributes. Such type of pharmacophore is a ligand based pharmacophore, and which I have shown you here. It is a structure based pharmacophore where you have a protein and a binding pocket, and you are using the protein, binding pocket and ligand both together to identify the database. Let us see one more pharmacophore. This is the same I have done using Schrodinger. Now see how many types of pharmacophore models are there, that is using multiple ligands, which I was discussing right now. You can even have single ligand, you can receptor ligand complex, your receptor cavity, protein residues, and merged hypothesis. What is a merged hypothesis? Is the hypothesis which you create by ligands, that is ensemble of ligands on one side and a protein on another side, and you combine those, that is called as a merged hypothesis. So here this hypothesis is clearly shows my protein so this is my pharmacophoric futures. So you are defining the what is the which cavity you have to be in you have to say inactive spaces. No, now I'm not showing any inactive spaces. So this is the hypothesis settings. So if you are getting any, these are the acceptor, this is a donor, this is any the scoring functions, you are excluded volumes, you can exclude something. So this is what this hypothesis, if you click, and if you are ensemble of hypothesis, this is a hypothesis you're showing here. So this is your one more, this is two, this is three, and this is the four. These are the common pharmacophore features, which are the minimal pharmacophore features you are required for any creation of any, what do you say, pharmacophore model of for this protein. This is what is being uh, displayed here, and this you see this pharmacophore how it is identifying the ligand. This is a basic simple pharmacophore model. This is a auto pharmacophore feature is also there here. So you just close this, or you can even exclude. So now you just click. This is your pharmacophore features. So these are the basic things of that pharmacophore, how they are present, and what are the features they are adding, what is the distance conformers and everything. So now once you prepare this, you just click onto the ligand and you click onto the pharmacophore. So this just click your ligand and fix in your space, and you click your hypothesis, fix in that space, and you go for protein space and you just click your ligand, and it exactly, if you click on L, it exactly fits into your database. This is the basic features of pharmacophore modeling, your structure based part of your design. So how to design, how to fetch your molecules, how to create a pharmacophore based upon ligand or structure. This is what is being of today's interest. Okay, I'm stopping here, and I'll uh, discuss the more in next coming lectures. Okay, thank you.